Shall I say that again?
Yes.
Well, thanks for coming to talk to us, John. John's going to give us a talk entitled "How Big is a Buttercup?" He worked at Aberystwyth University until 2016, where during his last year he established Botanical University Challenge, which was held at Kew. This year, the finals were held in Oxford, with around 30 universities from across the UK and Ireland taking part. So it's really grown. It's on YouTube, by the way. So if you want to, you can look at it and enjoy watching and competing against the best young botanists of the year. John left Aberystwyth to become the Vice Chancellor of the University of Natural Resources in East New Britain, Papua New Guinea. He says life in Papua New Guinea can be awkward, but botanically, it's out of this world. He's worked in other tropical rainforest areas, and they just don't compare to the diversity he saw there. The book he wrote, The Frustrating Flowers and Puzzling Plants, is a guide to difficult plants in Britain. So I'll now hand you over to John, and he can start his talk. John, can we share your screen?
We'll do that.
That's it.
Right. How's that?
That's it. Yeah, I can see it.
Yeah, that's it. Perfect.
Yeah. Okay.
Yeah.
You can hear me clearly and you can see the slide.
Yeah.
Yeah. Okay. Right. Well, let's kick off then. Yeah. Thanks for inviting me to talk tonight. And if I put in too many plugs for the book, just give me a shout and say that's enough of the hard sell. So I'm here to tell you about this, the problem of measuring plants and why we're interested in doing that. So this is what I'm going to cover, is that the how and the why of we measure plants, and how do we, and really to an extent, why do we bother to identify different species? And then finally, I want to sort of get you thinking about, could things be improved? Are we doing things as well as they might be? So the first thing that I sort of started with thinking about is that botany is meant to be a science, and science uses the scientific method, has done since the Enlightenment. And if you sort of look up a definition of the scientific method, it obviously starts with the process of observation and measurement. So it's really important to scientists that we're able to measure things. Sure. So how do we go about doing that? And when and where has it been done within plants? So here is a slide where I've looked at the three of the common butterflies in the British Isles that you'll all recognise. There's creeping buttercup, meadow buttercup and bulbous buttercup, and I've extracted from there the heights that are mentioned in Stace, in Phyta and in Bentham and Hooker and Ecoflora that I mentioned earlier, which is a sort of an online database of summary of all the information that we've got on plants. And you can see in Stace there are different measurements. In Phyta, there's sort of relative terms of short, medium, tall and short. And if you look closely in the start of Phyta, they actually do give you some measurements and define what they mean by short, medium and medium tall. So there are some measurements in there. In Bentham and Hooker, that's of classic, sort of more than 100 years old to flora now, we get these descriptions of seldom above a foot, variable in size, so that's sort of descriptions that are not particularly sort of helpful. And I like the one is smaller than acris. So you've got to know how big acris is to know that bulbosa is smaller than it. And then in Ecoflora, we've got a range of different measurements given in there. And if you look in Stace, you often get a species height, and then in brackets afterwards, you get a maximum value. And at some point, I contacted Clive Stace and asked him about that. And he said what he does was to, whenever he gets a record of a larger individual of a particular species, then he updates it. So he sticks that value in there. It's the largest report that he's ever had of a species. So if it's in there as a description to help you identify it, I think it's more like a sort of a bit of a freak show or a Guinness Book of Records, and knowing that the greatest height of any human is probably not that helpful in terms of identifying an individual, but it's in there and it might have some value to somebody. But when it was well before I started to sort of think about writing Frustrating Flowers. You could count these as I go along, see how many times I mentioned the book. I started to wonder about the numbers that were in floras and wonder where they'd come from and what the sample size was and what it actually meant. And that's what I want to get you thinking about this evening, where these numbers are from and how they've been collected and what are their error terms and how helpful are they in the first place. So does it matter how big a buttercup is? Do we really need to know? Well, you might want to know, are they getting taller over time? As climate change is affecting our flora, we seem to be spending quite a bit of time worrying about how that might impact on distributions or how that might impact on flowering times and interactions with pollinators and things like that. But do we know if climate change is making our plants get larger and taller or smaller and shorter? And if we've not got a decent measure of how tall they are in the first place, how would we know if climate change is making them get bigger? And if you're looking at a flora, do we assume that a buttercup is the same height in England and Scotland and Wales? And if it's not, if they're smaller in the north of the country or larger in the north of the country, do we need to know that information? Would that be helpful? Is it sort of vital if you're sort of trying to identify them? And are they the same in different habitats? If you've got a buttercup in a grazed pasture or a tightly regularly mowed pasture, it's likely to be much shorter than one that's sort of cut for hay and for the most part of the year is not cut or grazed. So yeah, they're obviously different, but you don't get that information when you go to a flora and you just look up how high is that plant. And is that information really useful in terms of helping to identify them? Well, perhaps it is and perhaps it's not. Let's think about that some more as we go along. So we all know that plant height is really variable, and it not just depends on grazing, as I mentioned earlier, but we know that we can bonsai trees, and this is the same species of sort of pine there, just as a few centimeters tall and growing in the wild in a sort of deeper, more fertile soil, and it'll be several meters tall. Having said that, generally speaking, if you can recognize a plant, you're able to identify it, even if it's growing in a tiny crack in a pavement and it's just a centimetre or two tall, or if it's growing in a fertile garden plot, then there are no weeds or anything else competing around it, and it's 10 times as tall. You usually don't have any problem identifying it, but yet when you look at a flora, we're always sort of find that the plant height is in there, however flexible it is and how sort of uncharacteristic it is of that species. If you think about the information that we've got in terms of plants, you could sort of think across to other disciplines. So here's something that you'll recognise, even if it's from a long time ago at school. You've got a periodic table. And in this periodic table, in this particular version of it, you can see that we've got some some elements that are identified as gases and some that are identified as liquids, just a couple of those, and the majority of them there that are solids. Now, we know straight away that we can sort of change things from solids to liquids and gases by altering the temperature and altering the pressure. So when we provide this information, it's only assuming that these elements are at standard sort of pressure and temperature. Now, we could do something similar with the plant world. But is there an equivalent of standard temperature and pressures for plants? You'd have to sort of say, well, if we're going to grow this plant, we're going to grow it in a large pot, whatever that is for in terms of a tree. We're going to grow it in a standard fertility, and we're not going to mow it. We're not going to manage it in any shape or form in the way that they might do in an arboretum with trees. We're not going to have any pruning, any sort of damage. And it's just an example that you're aware of, even if you've not sort of thought about in the past. We're looking at sort of cowslips, obviously grown in a flower bed where it grows huge and produces loads and loads of flowers. And when you find it growing in a low fertility limestone grassland, it might just be 2, 3, 4, 5 centimeters tall. And as I mentioned before, you'll instantly recognize that this is the same species, but knowing how big it grows in the flower bed under these lack of competition, high fertility conditions, does that really sort of tell us anything? Is it useful to have that information? And certainly those genes that have evolved in that plant had probably never in the entire evolutionary history of the cowslip has it encountered conditions and environment like that. So the phenotype that you're looking at is a completely artificial one that the species has never experienced until the, sort of the invention of, sort of horticulture. And yeah, it might be interesting to know, but does it really take much useful as botanists? And I've mentioned this sort of example, if you like, already with sort of buttercups and whether they're grazed or just ungrazed. The same species can be hugely different in its height. So if we're going to record the height of plants, do we need to specifically say under what conditions they were recorded? And none of that information is currently available. And here's another, this example goes back quite a few years now, that got me thinking about these sorts of questions. So this is the NVC community W10 Oak Woodland, and it also happens another opportunity to plug Botanical University Challenge. This is the rather magnificent trophy that we award each year now for Botanical University Challenge, and it's crudely based on the species that you'd expect to find in a climax oak woodland in the UK. And so the trophy is made out of the species that you'd expect to find there. As you ascend the trophy, it increases with height, and the different woods are in the proportions that you might expect to find. So the common species have got more timber in the trophy. So at the top you'll see there's oak, and at the bottom you've got the birch species present there. And there's a little seam in between the two, and that represents ivy. And you might recognize yew in there, and there's some lime. So there's a different species. Now, the reason that I mentioned this in terms of tonight's talk is that I started to wonder some time ago about, are there rules that govern which species could coexist together? These are just not random collections. We know that from the MVC, that you don't find trees randomly in any woodland. It's the same species, well, in any plant community you expect to see together, and you regularly expect to see certain species more abundant and other species less abundant. So what are the rules that mean that species coexist, that can coexist and can't coexist? So I wondered about something called Hutchinson's rule, which is not widely known. The Hutchinson's rule says that various things that are similar but different are how different they are regulated by this ratio of 1 to 1.1 to 1 to 1.4. So if you look at the size of bike frames, for example, you find small bikes and then some medium-sized bikes and large bikes, and they are roughly in Hutchinson's ratio sort of numbers. And the reason for that is that if you try to market a bike in between the bottom two sizes, then parents are going to size, actually little Jimmy or Jane or whatever can get by with using that smaller bike for a bit longer. There's no point in buying them a new one. We'll wait until it's really awkward for them. So that there's not really a market for one in between. And similar rules apply in the music world, in the families of violins, violas and double bass, etc. And they also apply in recorders. If you sort of remember back to playing recorder at school, you get recorders in different sizes, and the differences in those follow Hutchinson's rules. So I wondered, going back to the W10 for a minute, if you were to look at the ratio of these different species that coexist, do they obey Hutchinson's rule? And that got me to looking up the heights of trees. And the height data that I found in different flora was vastly different between different floras, and there was no information there at all about the heights of trees in different plant communities. So the height that you get assumes that an oak is exactly the same height, or can be, in any different woodland community in which an oak appears. So it did appear that these species were obeying Hutchinson's rule. So I sent the paper off to a reviewer, and very wisely the editor of the journal said, well, have you tried just looking at random collection of trees rather than ones that are in plant communities, and test to see if they obey Hutchinson's rule as well? And lo and behold, they did. So at that point I realized that I was barking up the wrong tree, sorry for the bad pun, and threw that as a particular paper in the bin. But it was an interesting aside, but it did get me thinking much more deeply about the data that in floras and where it comes from and what it actually means. So in real life, it's very rare that we'd use a single trait like height to identify a species. Generally, we look at multiple traits, and often we'll do a multivariate analysis if we're interested in sort of defining the species and recognizing the species in the first place. So we'll look at multiple traits all at the same time. We'll not just go on a single one. So here's an opportunity. Obviously, it's difficult to do audience participation, but I'd like you to do a little thought experiment with yourself. If you've got some paper and a pen, you might want to sort of just write some notes for yourself. All of us biologically have two grandmothers. Now, many of them are sort of long gone, and many of us might have known them, but many of us are probably lucky enough to have known two grandmothers. Or if you can't think of grandmothers, most families have got a pair of twins in the family. And we might not think necessarily that we're naturally good at ID, but if you think about it, we are IDing complex characteristics all the time. So if your two grandmothers were to walk into the room, or a pair of twins to walk into the room, if you know them, you don't have any problem identifying them. You can instantly tell one grandmother from the other. You can instantly tell one twin from the other. It might be a bit more tricky with twins, but it's not something you don't have to get out of flora and key them out and measure the heights and measure the hair color. You just instantly know what it is. But if you were to describe your two grandmothers to somebody who'd not recognize them, how would you go about giving that information to somebody who didn't know your grandmothers or your twins? So if they walked into the room, they could identify one from the other. Right, so I'll just give you a little second or two to sort of think about that and perhaps write down, how would you go about telling somebody who didn't know your grandmothers, how would you tell them apart? How would you recognize them? What information would you give them? Right. Okay, that's enough time for that sort of thought experiment. And I'm guessing that probably what you've done is something like this. You've got young granny and you've got old granny. You've got tall granny and you've got short granny. You've got grey granny and you've got long-haired granny. You've got Welsh granny and you've got Scottish granny. You've got one who's a bit chubby and you've got one who's a bit sort of like she needs a decent dinner. You've got one granny who looks after a few chickens, and you've got another granny who used to be a scientist or still is a scientist. So you've got a mixture of traits. But if you think about what I've done there and what I'm guessing you probably done, it's relatively easy if you've got the two of them together, that one of them is taller or one of them's got longer hair than the other. But if only one of your grannies was to walk into the room, it's a much more difficult job to identify which one it is if you've never met them before and you've only got one of them in front of you. And that's the problem that we have with writing floras, is that if you've only got ranunculus repens in there, I can't remember which way around it was in sort of Bentham and Hooker, but to say it's smaller than the other one and the other one's not in the room, it doesn't really help you in a flora saying it's smaller than the other, or it's tall. Because how does tall, unless you're like using fitter example, unless you've got a really good description about what tall means, and tall might mean something very different to a tree than it does to a grass. So we've got all of these traits, and it's not always that easy to do. However, if you can identify your grannies, as soon as you can do it, it's dead easy. So we've got a mixture of quantitative, qualitative traits, but a lot of these are relative things and are much more difficult to use when you've just got a single individual that you're wanting to identify. So if we go back to those three species that we started, we've got ranunculus repens, creeping buttercup, meadow buttercup, and bulbous buttercup. I've then extracted the data this time from STACE, and I've looked through, I've not put the numbers in, but what I've done is to say, well, have we got this information have we got all the species for all the data for all the traits for all the species? And very quickly we see that we haven't. We've got plant heights in for all three, but the descriptions tells you that repens is stoloniferous, but you're left to work out for yourself that acris and bulbosa are not stoloniferous. There's no mention of stolons, but it doesn't definitely tell you not stoloniferous. There's just no description. And with bulbous buttercup, not surprisingly, it says it's got a bulbous base, but it doesn't mention the base, any information about the base of creeping buttercup or meadow buttercup. It doesn't say they're not bulbous. It doesn't say anything at all. It mentions with repens and bulbosa that you'll know that there's a little leaf stalk in the middle of the leaf, and there isn't in acris, but it doesn't say that there isn't a leaf stalk. It just leaves you to work that out for yourself. So if you want to extract information scientifically across the board for all of them, what we've not got is a standard set of information for all of the species collected in the same way. And as I've mentioned earlier, you've got no idea where this data has come from. So we've no idea what the sample size was. We've no idea when it was recorded, who it was recorded by, and the conditions it was recorded at. So we've got no means, we've got no standard deviations. We've just got sometimes ranges and sometimes numbers. And the one example there that we've got that's highlighted in red is sepals being recurved or not. So it does actually mention for the other two that the sepals are not recurved, but in the other cases you're just left to work out for yourself where there's missing descriptions. And there are some characteristics that are really much more helpful than others in identifying plants. So leaf morphology, leaf shape, can be a really important, really useful trait in identifying plants. Unfortunately, that just means that you get into really complicated language, which is not particularly great for the beginner. You tend to pick these things up relatively quickly, I suppose, but the terminologies can be a bit uninviting. But what's difficult about these is the scientific measurement of them. We can describe them, but if we want to sort of say how much like this particular leaf form is it, or what proportion of the leaves on the plant are this, we don't get any scientific ability to measure these leaf forms. We don't get a degree of ovalness or sort of lanceolateness. It just says it's lanceolate. It doesn't say it's 95% lanceolate. So it's just a description. So they're great at one point, but they're not that particularly sort of easy to put a number against. And here's another example, which is we use remarkably often in flora. Will say this plant smells like, and it often amuses me, it'll say things like, I don't know if it does, I've not checked for this one for wood garlic, but there are a number of plants like wood garlic, and it will say it smells like garlic. Okay, if you know that it's garlic in the first place, then why is that in there? And smells are very personal things. I don't know that you smell things in the same way that I do, and we often know that sort of there's genetic variation within humans, so only certain humans can smell violets, for example. And so big chunks of the botanical population don't have the ability to smell violets, and yet the descriptions still include how they smell or what they smell of. And these are interesting traits. They're nice traits. It's nice information to see there, but it's not something that's easy to sort of measure and quantify. And therefore it's not something we can say, is the smelliness like the plant height changing over time? Are buttercups becoming sort of more or less smelly than they used to be? And I think anecdotally, there's a story that monkey flowers, when they were first introduced to the UK from America, used to be fragrant and smell. But whatever maintained that sort of the selection for smelliness in the States has not here in the UK, and that over time the monkey flower mimulus has lost its smell. I don't know how true that story is. I've never had much fun of success in verifying it. Right, so the other characters we know that are much more reliable and robust. So even sort of anecdotally, we use expressions like two peas in a pod, because peas, whatever the conditions the plant are growing in, tend to be very similar in size. It's one of those characteristics that varies massively between species. So the smallest seeds you'll know are things like orchid seeds, which are illustrated there, all the way up to sort of coconuts. So there's a huge range of variation between species, but yet within a species, there is very little variation in seed size. So in plant height and leaf size and flower size, all of the traits are highly variable, but seed size is really constrained. So the image that you're seeing there of beans hanging from a tree, many of you no doubt recognize that's the carob. And the reason that I've included that plant on this slide is that carob is the origin of the word carrot in terms of jewelry. And the origin of that is that the seeds of carob were so uniform that they were used as a measure historically. And you could imagine if you're actually measuring precious metals by weighing them against a bean, then it really had to be a reliable characteristics, because you didn't want somebody using a big bean and then somebody else using a small bean, because you were being sort of cheated out the amount of gold that you were trading. So within a plant, within a species, there is very little variation in this characteristic. So it must be a much more reliable character to include in terms of identifying a species. Now, okay, the seeds are not always present when you're looking at a plant, but it's remarkable how little seed information is present for most species in most floras. It's there sometimes, but it's pretty unusual to find the seed sizes for large groups of plants, but it might be more reliable. In contrast, you nearly always get flower colour information. And we know that plants can be highly variable in flower colour. There's a sort of a well-known example of a colour polymorphism in the British flora. But what you might be less aware of is that nowhere is there an approved list of, or a verified list of which plants in the British flora have variable flower colour. So if you look through a flora, it will virtually always tell you the flower colour, and sometimes it will anecdotally mention the fact that it occurs in white forms. And so I've seen in floras, for example, that it will say creeping buttercup sometimes occurs in white. Now, I don't know about you, but I've never seen a white buttercup. I think I might have occasionally seen a white Calthopalustris, but never a buttercup. And other species, which I regularly see as colour polymorphic, there's no mention at all in the flora that there are multiple colour forms. So we've not got nobody's gone to the trouble of standardising the recording of this, and it's present in our floras. But we do have this rather eclectic mix of different things that we measure. We measure quantitative traits, we mention qualitative traits, and we nearly always include a distribution, even if it's just a description of the distribution. So it'll say found in the west of the country, like that. I can't even remember what the species is that I'm sort of illustrating there. And that becomes almost a self-fulfilling sort of trait, and it could be a bit of a problem for sort of recording, because if you're using its distribution to help identify it, then if you're outside the distribution, you'd be a lot less confident in saying, actually, I really think this is this particular species, but it can't be because it's not growing here. So we've included traits, and I think we've probably, to a great extent, it's been a little bit random in terms of which traits we've included and which we haven't. We tend to try and identify the things that are most useful in terms of identifying, but nobody or no group of botanists have ever sat down and said, we really need to record all of these traits for all of the species. And once we've got all of this information for all of the species, then it won't just aid identification of the species, it might aid doing all sorts of science, botanical science, and asking all other sorts of questions about the data. And when you find these terms, you often find fudge words. I mean, we all get frustrated by fudge words in floras when we're sort of looking things up and it says sometimes or occasionally or maybe and things like that. There are lots of fudge words that occur. So when it says against a flora, in a flora, this species is sometimes hairy, you're never quite sure, does it mean one or all of the examples that I've got listed? So does it mean that it's hairy when the plant is older, or is it hairy when it's younger and those hairs are sort of lost as the plant ages? Does it mean some individuals within a population are hairy and other individuals aren't? So if you're just looking at plants in a population, you might not expect them all to be hairy. And if so, is the majority of them hairy, or is the majority of them lacking hairs? Is it that they vary between populations or between different communities? Is it that the leaves at the top of the plants are hairy or the bottom of the plants are hairy? It's really quite unusual to get that information, and often we've got it, or sometimes we've got it, but it's again, it's not been recorded in a systematic way. It's not been recorded in a standard way for the entire British flora. So when we got that information, we really are guessing, what does it mean when it says sometimes? And there could be really useful information that we're missing out on, or it might not be, but we just don't know because it's never been measured in a sort of scientific way. Now, the next one example I've got is just to say sometimes there are examples of species where vast amounts of information are recorded and are available freely, usually on online databases. And the example I'm going to use is something from a former life. You got in the introduction to what I've done with my life, the fact that I've worked in various tropical countries. Well, in the early 1990s, which is when this group of trees were planted, I was working at the International Cocoa Gene Bank in Trinidad, and there are lots of gene banks around the world. I've also worked in a grass gene bank here in Aberystwyth, where we have large stores of ryegrass seeds stored away in vaults. But all of these different accessions, all of these different genotypes of cocoa in this example are grown, and they're grown in standard conditions at standards of distances apart, and they're looked at in this standard way. And just about everything that can be measured from these plants is measured, and that information is replicated over a number of individuals, and all of that data is available. And often we've got no idea why they're doing this. So for example, if you look at the cocoa example, the length of leaves, the length of the petioles, the length of the pulvinus, the width of the leaves, the color of the leaves, the number on the density of leaves along a stem, all these sorts of things are recorded. The pod colours recorded, the number of beans, the dimensions of the beans, everything that you can think of is recorded, and sometimes is used potentially by plant breeders, and it's collected for vast numbers of different genotypes. So a few species, we've got everything that you could possibly measure, even if we don't know what we're going to use it for. So the next thing I want you to sort of think about is the use of this information in terms of identifying it. Now, there's a diagram there that you may recognize with Darwin's handwriting against, which is his famous little first thoughts on evolutionary relationships and branching trees. And for a long time, that's the way that we thought about evolution working, that species split from one another and they split again. But we now know that it's not uncommon in plants for them to hybridize, and to hybrids then to become one way or another a new species. Now, the reason I've also inserted into this image a picture of radiate groundsel is that that's what I did my PhD on more than 40 years ago now. And what I sort of found when I was looking at that, radiate groundsel was much more common back in the 1980s, which is when I was looking at this, than it is today. And what you find when you look at radiate groundsel populations across the British Isles, that wherever you look at groundsel populations, there appears to be hybridization and introgression back into groundsel from the local Oxford ragwort population. So wherever you look at groundsel, radiate groundsel within that population was more like the local Oxford ragwort than it was another Oxford ragwort. So in everywhere around the country, there was hybridization going on and back crossing. So those species are clearly distinct. Nobody had any problem identifying them, but there was merging at a local level of these two genomes. And something else that I'm going to mention at this point, and I'm aware that Mary Dean is sort of listening to this talk. Hi, Mary. Is that Paul Ashton, who's the incoming president of the BSBI and a good friend of Mary's, also worked on the same problem at a very similar sort of time, and has a similar view about the hybridization as an important role in the speciation of plants, particularly because Paul was supervised by Richard Abbott, who sadly died this last year. And I've been talking to Paul recently at the last Botanical University Challenge, and we both independently, I think probably at Richard's death, been thinking about the importance of hybridization, and perhaps we should think again in terms of the implications of that for species identification and the tools that we use. And Paul had already come up with a title. We thought we should write something for the BSBI newsletter or something, and Paul was wanting to call it the tyranny of the trees, is because we've got this traditional way of thinking of dichotomy, is that even the software that analyzes molecular data tends to put them in the species into bifurcating trees, in spite of the fact that we know that at least half the plants on the planet, half the species, have got a history of hybridization in their evolution. So we know that things rejoin, and yet almost implicitly from that thinking, we use dichotomous keys. Now, here's another opportunity for me to sort of put a plug in for my book, which is very reasonably priced and available on Amazon. Or you can, I should say, you can get a discount if you're a BSBI member. Is that I took a different approach for a number of reasons, mostly because of a lot of these complicated groups. The reason that they are complicated is that they've got hybridization involved either recently or more historically in the creation of a gammosperma species. And often you don't get all of the information present for all of the species. And sometimes because they're not in flower, or the leaves have fallen off, so you've not got all of the information. Where if you've got a multi-axis key, a tabular key, you can sort of tick off the information that you've got, and then you can look over all the species and say, well, what's the best fit over all the information that I've got? And I had quite an interesting conversation following the publication of this with Arthur Chater, the wonderful Arthur Chater, who happily for me also lives in Aberystwyth. And Arthur said, I've probably produced more dichotomous keys than anybody else in the British Isles. And I don't think I ever had the thought in the back of my head that it was related to the evolution of the plants in the first place. And I said, no, I don't think we do, Arthur. I think it's just part of the way that we think. We don't have to think about it while we're doing it. It's just the way that things have developed. So we do it automatically, assuming that that's the best way to do it. And it works. Dichotomous keys are sometimes great, but we all know that dichotomous keys can sometimes be really frustrating. And one of the reasons that they're frustrating is you run into problems like this. So there are 4 species of red dead nettle in the British flora. And if you work through the dichotomous key in Stace, you will get to a split that it says teeth more or greater or less than 2 millimeters, right? And then you think, well, yeah, I've got that information, but it could be one of 4 species. So it would be useful to know what is the tooth size of the other 2 species. And just as we saw with the buttercup example in Stace, there is no information. It's just not there. So if you wanted to say, well, what about the teeth in the others? Are they smaller or larger than 2 millimeters, or are they just absent? Is it just one of these gaps? Their teeth are not present? You don't know. There's no information there whatsoever. So what I found when I was trying to populate these tables in my flora is that often if I'd got a group of similar species that you might want to compare, I was wanting to make sure that I'd got all the information for all of the species. And often that is just not available. Now, sometimes it was relatively easy to fill in those gaps and say absent, and sometimes it's not. But lots of the times that information on the measurements of traits was just not present. And it's a huge task for one individual to go and start measuring and filling up all the gaps in all the floras. So sadly, what that meant is often I had to leave out some particular characteristics, and I tried to include things which were simple and easy to understand for relatively novice botanists. And on occasions, I was forced to go back and actually sort of measure some to fill in some of the gaps. Now, there quite legitimately, if it was a real audience sat in front of me rather than a virtual one, you could be shouting out, well, how did you do it, John? What sample size did you use? What time of year did you use it? Where was it? What was the mean? All these criticisms that I've been sort of pointing at other floras for not including, I'm equally guilty of, and that's because it's a really difficult task to get all of that information for all of the species. So when I've filled in gaps, I've really done it for fairly crude traits. So I've not given precise information of the height, for example, would be between 2 centimeters and 5 centimeters. I've said it will be greater than 10 centimeters, really, if it's a species that is regularly much greater than 10 centimeters. So I've only filled in these, provided this missing information where it's been absent and where it's been pretty clear that what the missing data would be sort of like. So it's not precise data. It's just, yeah, these are really big or really small, and I've given numbers to that. So let's think about these problematic species that I've been focusing on in frustrating flowers. There are certain groups of plants that are much more difficult to identify than others, and that's was something that I sort of recognised that there was a gap in terms of publications. There are the wonderful BSBI handbooks, but there's not just a general guide, and I sort of spotted that when I was chairing the BSBI committee on sort of education and training, as it was called at the time. And if you start to sort of think about these particular difficult groups, you then need to say, is it the same characteristics that we need to be looking at to help identify these, or do we need to be doing these in a different way? And there are also questions about the level of resolution that you need to go. Now, I've included apples on this slide just to get you thinking about the taxonomic resolution that we need and how good an identification it needs to be. And shortly after producing this book, I was sort of teaching a course for the FSC and Manchester Met on identification of difficult plant groups. And the person I was co-teaching with, Mark Duffel, that some of you will know, introduced me and said, John, definitely a lumper when it comes to difficult groups. And I said, hang on for a minute, Mark, I don't think I am. I don't think that that's what I'm trying to achieve. And I think apples are a great example of this. You might never have thought of apples in these terms, but different apple varieties, many of which you'll recognize straight away, are effectively apomictic species. They are they breed through, all right, the way that they breed is via humans taking cuttings and replicating them, so that the human has been hijacked by the plant, we've been domesticated yet again by a plant. So it's rather than a dandelion producing asexual seeds to reproduce, these apples are much more successful vegetatively at spreading around the world, but they're getting us to do the difficult work. And sometimes that really matters. Sometimes you want an apple and it has to be an apple of that particular variety. So sometimes you just want, in this case, we've got a Cox's orange pip in there, if you recognize that. And other times, if you're making a pie, you don't really care what variety is, but you might want to know that it's a cooking apple rather than an eating apple. So I've got 3 apples there and a drawing. So sometimes you might want to know the absolute variety. Sometimes you might want to know, is it a cooking apple, is it an eating apple, or is it a cider apple? So the bottom one's a cider apple variety. And sometimes if you're teaching the kids their ABCs, then it doesn't matter what level of resolution you go. All that matters is A for apple, that they can identify an apple. So sometimes I could say there are good reasons to be splitters, and sometimes there are good reasons to be lumpers, and therefore the level of resolution that you need to go to and the complexity of the traits that you need to think about is dependent on the question that you're asking in the first place. Why am I wanting to identify that apple? Am I going to do it because I want a particularly good eating apple? Am I wanting to do it for something just cruder? Am I going to make cider with it or make a pie? Or am I just doing it so I can teach my young kids how to identify an apple or how to sort of recognize the letter A? And that's something that I've tried to sort of get people to think about and have covered within the sort of flora. And I've broken it up roughly into these 5, can't count, different reasons for complexity, which as botanists you'll recognize instantly that the sort of plants that are difficult to identify tend to fall into 1, sometimes 2, sometimes even 3 of these groups. So there are things which are asexual, so we get the dandelions that I've mentioned already, so there can be hundreds of dandelion species that can be tricky to tell apart. There can be species that hybridize frequently and that tend to merge, and it's difficult to sort of recognize one from the other. There can be groups of plants that appear to be rapidly evolving from one species to another, so it's not always clear where they are in that evolutionary process. There are other groups of plants that inbreed regularly, so different populations can differ from each other just by chance of founder effects. And possibly most puzzling for novice botanists of the lot, there are certain families that, like the umbels there, like the Apiaceae, that they are if there were just 1 or 2 of them, they wouldn't be difficult to identify at all, but they become more difficult just because there are lots and lots of them. So there are lots to wade through and lots to become sort of familiar with if you're a sort of a novice botanist. So I broke the sort of group plants up into these different groups. And as we mentioned earlier, we tend really scientifically to use multiple traits to identify an individual to one species or another. And even when we do that, we'll get some individuals that don't neatly fall into one species or another. So when we look at multiple traits, even then, it's not always definitive what an individual is. Sometimes we'll just sort of say, well, it probably is a hybrid, but it looks like it's got sort of characteristics, or it's more like this one than it is the other, but it's still got characteristics of both species. Now, to an extent, it's getting simpler with DNA barcoding, and we are really lucky within the UK to be the first nation on earth to have its entire flora barcoded, by which we mean sort of sequencing key bits of the genome of all the species of plants in the British Isles. It's not particularly great as a field identification trait, but once you've got the ability to identify with some scientific rigor by looking at the DNA, by looking at sections of the genome which don't vary very much, then we can be much more confident that we've got characteristics that mean that we can be confident about how we're identifying a species. Now, the bad news is that if you look at the British flora, there are pretty equal numbers. There's 45% actually. I've got this data from Tim Rich, who used to sort of teach the same difficult plant group before me. So this is a slide that I've borrowed from that presentation. And of the plants in the British flora that we've barcoded, we've really done what he described as normal species rather than the apomictic one. So they're really difficult to identify things. We've not got that barcoding information as yet available, and we're probably going to need to look at different parts of the genome and going to need higher levels of resolution to sort out to these highly complex asexual species. But what it might be interesting for is for admitting what we very rarely do, is that we assume that these the great and the good, the best botanists in the land, just don't make mistakes, and that we send a plant off to the authority for that species, or to we'll have a discussion with our county recorder. We'll expect that they'll get back to us and say, yeah, you're right, it's this species or it's that species, and that they never ever make mistakes. Now, the ability to barcode means that we can actually, for the first time, start to test that. Now, the reason that some botanists are suggesting that we should be doing this is not to embarrass sort of these sage old botanists who've been doing it for a year and question their botanical skills. Is that the reason that we might want to do it is that if we were to give some complex plants to different experienced botanists and then say, please could you tell us what this is, and then check that against the DNA barcode, we could then look at the proportion of times that they make errors. Now, that is not necessarily interesting in itself, but if you were then to sort of do that over different groups of plants and say, actually, people, our expert botanists, are 99% accurate or 99% consistent with the DNA barcodes for identifying members of the Asteraceae, but when they come to the sedges, again, one for Mary and sort of Edge Hill sort of connections, actually, even the experts get the sedges wrong 20% of the time, then it's really useful to know that the sedge keys need more attention or the botanists need more training. But at this point in time, we've not got that information. We just assume that all botanists are equally good at identifying all species. But now, for the first time ever, we can start to question that, and that might be a useful thing to do in terms of the certainty we are of botanical records. So in summary, the best studied flora on the entire planet, certainly in terms of distribution and change of distribution over time. I don't know if you've worked out what these species are, but again, it's the same 3 species of butterfly that I've been talking about throughout. But getting to the end now, we're doing for time. Yeah, I've been talking for 55 minutes, so we're getting on. So what I've been sort of trying to get across is that historically, botanists have never scientifically measured these traits. We've never said they are measured from 100 plants. They've been recorded at this place, at this time, by this person, and this was the plant community that occurred in. And not only have we not done that, we've not recorded the same things for all the plants, and we've not really sat down and scientifically given that much thought to how we should do it. In fact, if you were to ask 3 botanists how big is a buttercup, you'd probably get 3 different answers. So here you've got 3 different botanists, and they are not 3 different botanists chosen at random. There's Alistair Fitter there, who was my PhD examiner many years ago, and who started Ecoflora database. And then we've got Henry Ford, who's helped run Ecoflora database. If you don't know, it's an online suppository of all information that we get on of plants. And the 3 of us would all agree that we might not agree about how big a buttercup is, but we would all agree that we need to do something about it, that it needs to be measured more scientifically, and we need to think about how we're doing it. So one positive note to end on, we could take on this task as a national group of botanists and send out people to record it. But there's within Aberystwyth, there is this setup, which is a national resource. So this is the Plant Phenomic Centre, which is a huge number of greenhouses which have got those standard temperature and pressure conditions that I mentioned earlier. Now, we've never agreed what they are for the plant world, but we could do. So plants are grown in pots in uniform conditions. Everything is controlled by computers so that their environment is completely regulated, and every single thing about those plants is recorded, and it's done via a whole range of different sensors. So it's not just done by light photography, it's done by bouncing all sorts of different spectrums off them, and it's recorded automatically and captured. So we could, if we wanted now, for the first time ever, scientifically record all of these traits. But really, if that's going to happen, it's going to take a research council to fund it, and it's going to take somebody to recognise that we've got a problem. So there we go, folks. Thanks for your time. Thanks for listening, and I'm happy to take any questions.
Okay, thank you, John. You can stop sharing your screen now.
Yeah. Has that gone?
I can still see you. Oh, yeah, I can still, because of the speaker. Yes, it's gone. Yes, thank you. Right, any questions for anybody? I've had a few thoughts while you were talking.
Good. I'm glad.
Yes. I mean, you recommend recording every detail for every plant.
Not every detail. I think what we need to do is work out what we need to record and record the same things for everything. Not everything. We need to think about what we record and why.
Okay. I mean, some books are pretty good. I mean, Sella Morel is not perfect, but it's much more thorough than your average flora.
But yeah, it's true. Some are better than others, but we still don't know where it came from and how it was recorded and what the sample size was. And
no. And relating to sample sizes, I mean, the plants are very variable, and seeds are fairly consistent. But I was, for my own interest, I've been looking at plum stones, and I collected a range from a particular plant, tried to get them all from the same bush, and got them out, measured them, and they are pretty consistent, but there is a range. And
there is, you're right.
Yeah, and I wondered whether you'd have to do, and I took the average of all the ones I measured, but I should have knocked off the height, taken off the highest, the longest ones and the shortest ones, just so you've got a trimmed mean to take out the
But yeah, Stace records the largest. It doesn't record the smallest. But I mean, there is a case for recording the smallest, the largest, the mean, the standard deviation.
Yeah,
all of that information.
That's a lot of work.
It's a lot of work, but then we have got a go on.
Measuring seeds. I mean, plum stones and sloes were bad enough. They were about 8 millimeters. But in the field, if you were trying to measure your poppy seed, or your orchid seed, you might run into difficulties.
Mhm.
Another thing I was thinking of, you were talking about dichotomous keys aren't very good. There are multi-access keys, and a place I worked once had a system for identifying bacteria, and you have a list of characteristics, and each in triplets, and each one has a score 1, 2, and 4, and you write down a plus for the ones that exhibited that characteristic, and then you added up all the scores, and at the end you ended up with a 7-digit number.
Mhm.
And you could relate that to a particular type of bacterium. And I've often wondered if it's worth doing that with plants. I think it would. One thing I tried to do with Cotoneasters once was to make a row of plant characteristics in Excel, and then if it and from the list of species, and type in what they had, and then if sorry, I'm losing my way. Yes, and then you can filter each column to only exhibited the species for the characteristics that you found were there, and you ended up with just 2 or 3 taxa to sort out in the book. I don't know if anyone's done that, but it seemed to me to be quite a good way to do it.
The thing I mentioned sort of towards the end of the talk about once you've got DNA barcoding, you can actually test how accurate these things are. That applies to multi-access keys and the methods that you just described. So
Yeah,
that seems
yeah.
an obvious way forward, is just now we can identify these things much more definitively. You can test to see how good the different approaches are, and see which ones are more successful, can't you?
And
That's
how
the
does the
scientific thing to
barcode
do.
work? Remind me, how does
The
the barcode?
barcode sequences particular bits of DNA within the
Right,
plant's genome,
right.
usually related to a key function that selection means that it's not going to vary. So
Yes,
bits between genes vary vastly, so you get lots of noise, and bits of DNA that are really important are highly conserved. So if you're looking to identify things as a fingerprint, you want something that's highly conserved, and will be sort of definitely that species and not another. So they tend to look at these sections of DNA that are highly conserved, and they sequence them, and then they compare the sequences. So you've got,
right.
this is the sequence for this species, this is
Yes.
the sequence
Okay.
for the next one. So if it's got this sequence, it's X. If it's got the other sequence, it's Y.
So it's no use in the field.
It's no use. Well, it's not at the moment, but it'll not be long before somebody's
Oh,
sort of
oh
got
yeah.
and photo apps on phones and things are getting better all the time. They tend not to be
Yes.
so good at these complex groups, but they're remarkably good for identifying
I,
plants.
I'd agree with that. For all the things that you could do by eye anyway, they're pretty good. But if you get something that's difficult, um, they're likely to lead you down up the garden path. Anyway, is anyone else got anything they'd like to ask?
I was just going to ask something about which you sort of just about touched on, about which sections of the genome we look at when we do this sequencing. And obviously, there's been some decisions about which are most useful, but we've often speculated when we've been kind of out on field trips and things, if we looked at an entirely different bit of the genome, would we actually still be coming up with the same answer? So does it sort of go around in a bit of a cycle? We think these bits are useful, and therefore we have a system that works, and it works quite well based on them. But we could be missing possibly some other bits of the genome that we perhaps haven't figured out are useful, or are we beyond that? And there's obviously a great deal of variation between individuals within a particular species, which will be reflected in the genetic, which I guess is what you're talking about as noise. But it's kind of, particularly when we start going into things like subspecies, then it must be trickier, even with DNA, to really be sure that we're separating things out accurately?
That's a great question. And I think that at one level, we know the answer to that is that you will find a sequence and it will say this is different from that. But if you're looking at different sections of the genome, because of that history of hybridization, of which genes have come across sometimes and which one they haven't. You think about the groundsel example. The groundsel in Aberystwyth is more like the ragwort here than it is down the road in Shrewsbury or whatever. Is that if you're looking at different parts of the genome, then you'll get a different taxonomic tree, which is what Paul's worrying about with the tyranny of the trees and the fact that you're forcing it into this dichotomous taxonomic relationship. However, if there's hybridization going on, you're not getting these branches coming together and fusing and then splitting again, because it's not the way that the taxonomic software works. And so, yeah, I'm sure you're right. If we were to look at different bits of the DNA, we'd still be able to identify the species, but how closely they are related to each other, I think we'd get a very different picture. But we do, don't we? We know that we do.
Ian's got quite an interesting point in the question, George. Do you want me to, I don't know if Ian wants to kind of make the point himself or just want us to read it out. But
read it out, Maria.
okay, so he's saying with regards to the DNA barcodes in animals, the 658 base pair section of the COI mitochondrial gene, from his experience in moss species that are clearly different morphologically and ecologically, can share DNA barcodes. So he's saying in his experience that they recommend them as another data source to use in conjunction with morphological and other data sources used on their own. They actually can be misleading. I don't know if you've got any thoughts on that in terms of plants.
Well, my understanding, I've not really been there, in spite of the fact that the work was done at the Welsh Botanics, who I've collaborated closely with over the years, and here in Aberystwyth. It's not really sort of my sort of area. But yeah, my understanding is that they are actually much more of definitive in plants. But you're absolutely right. There are occasions where they don't resolve. But then if you're unable to resolve them, and there can be a host of reasons for that, then you just look at other sections, because yeah, if they are genuinely different, then they're somewhere in the sequences you're going to find those differences that are causing the phenotypic differences. But yeah, in terms of agreed sections that are looked at, and these things are agreed internationally, the 2-3 main set of sections that are looked at in terms of plant barcoding, they've been pretty successful, and that's not a huge problem.
Thank you.
Okay, thank you. Anyone else? Okay.
It's Graham's got his hand up.
There's Graham.
It was more of a comment on the DNA material. Sorry to this is stuff I teach, but I'm no huge expert. That with animals we use mitochondrial DNA because it has a high mutation rate. So we use a conserved gene, so the PCR primers work, and then we amplify the bit up in the middle. So one reason for using a conserved gene is that the primers, the technique is more likely to work, and then we're sequencing the bit in the middle, which is the unknown bit. The problem with plants is plants on the whole, most of the genome has a much lower mutation rate. So it's been really problematic. It's been really problematic to find regions of the genome that are variable enough. So in a sense, I slightly contradict what John's saying. In a sense, you want a conserved gene in plants to make the prime work, somewhere that's going to work universally across a lot of species. But you then need a gene that's variable enough to be able to distinguish one species from another. And there are lots of catch-22s there.
There are other problems as well. I mean, one of the problems there is they target, in the same way that you've been talking about mitochondrial DNA, they target sort of chloroplast DNA and sort
Yeah.
of and one of the messy groups that I've covered are the broom rapes. Well, don't have any chloroplast DNA, so
Yeah,
you can't use them in the first place.
but chloroplast DNA, in a sense, chloroplast DNA is an extra nuclear genome. It's outside and away from the nucleus. So it'd be as is mitochondrial DNA.
As is mitochondrial,
Yeah.
exactly.
So it's kind of the obvious one to perhaps try to use to do the equivalent for animals, barcoding in animals and plants. It's kind of the obvious way to go. But chloroplast DNA has a really, really, really slow mutation rate. It's been really, really problematic. So in that case, I could see that you could have a whole genera of plants, 20 different species, of which half the species would share exactly the same sequences, or you would have
it's that group
one
that I
or
mentioned,
two
the ones that are...
one or two mutations between species. It doesn't tell you anything, but also then within a species, you probably get one or two mutations as well. So you've got variability. You've got very little variability between species, but you've also got variability within species. So it doesn't really throw any more light on the. It doesn't really throw any more light on the picture.
Well, it's particularly murky with some as well, and it's murky with the rapidly evolving groups, because they just haven't had the time for that variation to occur with the slow mutation rates. And that is one of these groups of complex plants. And this, it's not a surprise that they've not barcoded these groups yet. And that's one of the reasons. They've done the easy species, but the complex ones that they're still struggling with. And another group, of course, are the hybrids. Because the hybridizations, you're going to get your chloroplast from one parent or the other.
Yeah. Yeah, no, I really enjoyed your talk. It really made me. And
the hybrids are often in the apomix as well, because that's often why they become apomictic, isn't it, in an evolutionary sense?
I teach this sort of stuff, and your talk really made me think. You definitely made me think about the way that I'll continue to teach something in the future. So I do genuinely really appreciate that. You really made me think about things. Yeah.
That's great.
Yeah, thanks.
It's always nice to think.
Yeah, no, brilliant. Okay.
Yeah, it's coming up to 8:15. We're running over a bit, so I'll draw the meeting to a close now, unless there's anybody has anything else they want to add.
Thanks for the invite, folks.
Yeah, no, thank you, John. That was absolutely brilliant. It covered a wide range of things, underlined a lot of problems, drew the distinction between vague things that we use in the field to identify plants just by looking at them, and actually measuring things and quantifying it, and making it more consistent. So thank you very much.
Thank you.
Yeah, thank you. And I just I really did enjoy that as well. I just want to mention that next week we've got the next in our series of AGMs. So do join us for that as well next Thursday. All the details are on the website, and just register and do come along for that as well, because I'm sure that'll be another really interesting session. Thank you.
Okay, thank you very much, and goodbye, everybody. I'll switch off now.